After studying about replication, let us start with the second process, that is transcription of DNA. Transcription defines the conversion of DNA to mRNA. Therefore, it can be defined as the mechanism or process in which the mRNA, which is called as messenger RNA, is derived from DNA which involves the formation of an RNA strand on the DNA template. This process of DNA transcription is almost similar to that of DNA replication. In this process, the DNA, by getting converted to mRNA, give its information to the RNA. Now, the process of transcription generally occurs in 3 steps. First step, the enzyme transcriptase gets attached to the initial region of DNA. That is, for transcription, this initial region of DNA is considered as a specific site for getting converted into RNA. This region of DNA has to be transcribed. With the help of enzyme called transcriptase. In the second stage, that is, after the enzyme transcriptase gets attached to the initial region of DNA and the 2 strands which are separated starts moving away from each other, following which one of the 2 separated chains of DNA acts as a template chain. It is this template chain which provides the information for synthesis of mRNA. Therefore, this occurs on the basis of nucleotides that are present on the template chain, as a result of which the enzyme transcriptase, it arranges complementary RNA nucleotide units one after the other. Thus, arrangement of complementary RNA nucleotides gets arranged one after the other and phosphodiester bonds gets formed between. As a result of which a new mRNA chain is synthesized. Which is followed by the last and the third phase, that is of completion. That is the completion of the information that is synthesized within the mRNA. In this, a specific protein attaches with the mRNA molecule and separates it from the template chain. This template chain involves both DNA along with the enzyme, that is transcriptase. Thus, in this manner, the new mRNA chain that is formed from the template chain gets completely separated from it and occurs as an individual new strand. After which, this newly synthesized mRNA strand gets translocated to the cytoplasm where it gets associated with the ribosome to form protein. Further, after association of mRNA with ribosome to form protein is described as another process, translation, which we'll study later. This figure depicts the transcription process, that is conversion of DNA to mRNA. As it can be seen from the figure, the DNA gets unbound and results into the formation of two individual strands, out of which one template strand results into the formation of RNA strand, which creates a RNA-DNA hybrid region, where this RNA-DNA nucleotide molecules joins with the help of phosphodiester bond and gradually gets separated from the DNA molecule and acts as an individual mRNA. Thus, in this manner, with the help of DNA gene, the transcriptase enzyme that becomes attached to form a separate chain, which further enters into second phase and forms the mRNA molecule in the form of complementary RNA nucleotides that joins with each other through phosphodiester bond. Following with the last stage, that is completion stage, occurs where the mRNA molecule gets separated from the DNA nucleotide to act as an independent material, where it gets separated from the template DNA chain and the transcriptase enzyme, which it further gets translocated to the cytoplasm, where it gets associated with the ribosome. Thus, in this manner, the transcription process occurs where DNA is converted into mRNA.